Today we're going to go over some information from Chapter 5, and then we will finish the presentation with a mention to your Pineapple Enzyme Lab that we will be conducting. Chapter 5 is about energy flow. Specifically, we are interested in energy flow within living organisms, or what we call bioenergetics. So bioenergetics refers to the energy flow and energy transformations that are going on within living organisms. One thing that you want to remember is that all living organisms must acquire energy from their environment. This is one of the characteristics of life discussed from Chapter 1 information. Let's make sure that we understand that the sun is the ultimate source of energy for all living organisms. I'm going to draw a basic diagram that shows energy flow from the sun through living organisms. So let's start with the sun. The sun radiates different types of energy. Some of the energy that radiates from the sun is heat energy, and other types of energy that radiate from the sun is light energy. There are other types of energy that radiate from the sun, but we're going to consider these two at this point. Let's draw a plant which represents a photosynthetic organism, specifically the leaf of the plant. The type of energy that the sun radiates or emits that is important for the plant is called light energy. Some of the light energy that the plant or the leaf is exposed to is going to be absorbed and used to produce an energy-rich molecule, which we'll call glucose. So the sun emits various forms of energy, and eventually that light energy will be trapped in the nutrient, which we're going to call glucose specifically. Other types of nutrients may be produced by plants, but we are going to use glucose as an example. So remember that bioenergetics is the study of energy flow, basically from the sun and within living organisms. So let's talk about where the energy is going to flow to next to that after the leaf. The energy will ultimately flow to animals and other organisms. And it flows this way when we basically consume fruits or vegetables or any sort of products produced by plants. We'll draw your dog or your cat here for good measure. The leaf represents a photosynthetic organism. Photosynthetic organisms undergo reactions called photosynthesis that ultimately produce nutrients. Producers end up making nutrients for all other living organisms on Earth. Whether you are eating your plants directly or you are eating an animal that has already eaten a plant or a plant product before you, all of these nutrients are coming from photosynthetic organisms by the process of photosynthesis. Since this is the group of organisms, and this would be example of animals, but we have many other organisms that consume the nutrients produced by producers, we're going to call this group of organisms the consumers. Consumers are going to intake the nutrients produced by the producers and break down all those nutrients through a process called cell respiration. When the consumers die, the energy is going to flow to a group of organisms called the decomposers. The decomposers will metabolize or break down the dead organic matter that was ultimately the product of the consumers. The decomposers ultimately break down the dead organic matter into simpler molecules, which are going to end up being recycled and usually recycled by the producer type organisms. As energy flows from one living organism to the next living organism, some of the energy is released. Energy is also released when the leaf converts light energy into the energy that will be trapped inside of the glucose molecule. The heat energy that is released is ultimately an unusable type of energy for living organisms. An example of an energy rich molecule that has usable energy would be glucose. So notice that energy is always flowing in one direction where heat energy is consistently released. Give you a chance to write all of this down. Let's continue on the next page. Let's talk about the definition of metabolism. Metabolism are all the reactions that happen in cells that use or produce energy. Sometimes you may have heard the terms someone has a high metabolism or a low metabolism, but let's recognize the definition. The definition of metabolism are all of the reactions that are specifically happening in living cells that are going to either require energy or end up producing energy. If the reactions are within cells and cells are living, we're going to call these biochemical reactions. Ultimately, there are two categories or major types of reactions that are happening inside of cells. The first one is going to be called anabolic reactions. Anabolic reactions are the types of reactions in which larger, more complex molecules are built. An example of an anabolic reaction that we have discussed previously is going to be dehydration synthesis. Dehydration synthesis refers to a specific type of reaction in which the larger complex molecule that is assembled is an organic molecule. An example of dehydration synthesis would be building proteins or building DNA or building complex carbs. So here are two examples of dehydration synthesis. Let's go over the basics about an anabolic reaction with a diagram. During an anabolic reaction, you start off with simple molecules, and after the reaction occurs, you build a larger, more complex molecule. starting molecules are called the reactants, and the arrow indicates that the reaction has occurred. The molecule that you end up with is going to be called the product. In this case, there's only one product. The other type of reaction that is ultimately the opposite of the anabolic reaction will be the catabolic reaction. Catabolic reactions break down large complex molecules into simpler molecules. An example of a catabolic reaction would be a hydrolytic reaction. We discussed hydrolytic reactions in Chapter 3. A hydrolytic reaction refers to the breakdown specifically of an organic molecule into smaller subunits. Let's draw a basic diagram to show the overall meaning of a catabolic reaction. Ultimately, it's going to be the opposite of an anabolic reaction. So we start off with a large, complex molecule, and the reaction will occur. We're going to call this a catabolic reaction. And we break that larger complex molecule down into individual molecules. The arrow will point to the products of the reaction. What you start off with is always going to be called the reactant. Examples of hydrolytic reactions would be when organic molecules like proteins or fats or carbohydrates are broken down during digestion. Let's talk about our next question called metabolic pathways. A metabolic pathway is going to refer to a series of biochemical reactions that convert substances into other molecules needed for life. Metabolic pathways can be complex. Let's write down the definition of a metabolic pathway. I'm going to draw a very basic diagram to show an example of a metabolic pathway. We're going to start off with substance A. That's going to be our beginning substance. And we're going to draw a series of reactions that occur in order to reach the goal molecule, which is going to be the final product of the metabolic pathway. A metabolic pathway is a series of chemical reactions that occur in order to produce a substance at the very end of the reactions, which is the final product. I like to also call this the goal product, since this is the reason that the metabolic reactions or the metabolic pathway occurred in the first place. So we're going to start off with this substance right here, which is sometimes called the substrate. And in order to get to the final goal product, we have to undergo a few specific types of reactions. So we're going to call this reaction number 1. Here's reaction 1, reaction 2, reaction 3. This diagram shows that there are 3 reactions that are needed in order to get to the final goal product. So, for example, when a plant is undergoing photosynthesis, its final goal product is to produce a sugar called glucose. And there are a series of reactions that take place in order to make that final product. These substances that are produced in between the beginning substance and the final substance or the final product are going to be called intermediate substances. At each reaction, it's important to note that we need a specific enzyme to drive that reaction. Oftentimes, metabolic pathways are made of 25 or 30 or more reactions. So this is a very simplified diagram of a metabolic pathway. And during every single reaction, a specific enzyme is needed for that reaction. For example, in order to get from A to B during reaction 1, we need a very specific enzyme substance called enzyme 1. Let's give 2 examples of metabolic pathways that we will discuss in more detail, and they're going to be important and absolutely critical for life. The first metabolic pathway is called photosynthesis. Photosynthesis is the type of metabolic pathway that requires a very large input of energy in order to make sugars. So the point of photosynthesis is the plant to produce sugars or various other carbohydrates. And since this is the type of metabolic pathway where the goal is to build molecules, this is going to fall under the category of anabolic type of reactions. The final goal is going to be carbohydrates. The example that we're going to use in our class is a type of carbohydrate called glucose. I want you to realize that not only glucose is made, but glucose is the most common type of carbohydrate that is produced by the photosynthetic reactions. So the photosynthetic reactions are a series of 20, 25, or 30 reactions in which light energy is going to be absorbed by the plant and then ultimately used to produce a high energy substance called glucose. So keep in mind, glucose may not be the only type of substance produced. There may be other energy-rich substances produced. You want to make sure you understand that glucose is a high energy molecule. The other metabolic pathway that is definitely important to life is called cellular respiration. During the process of cellular respiration, sugars are broken down. Sugars or other energy-rich substances are broken down. And anytime something is broken down, remember that we categorize that as a catabolic type of reaction. So remember that glucose is a type of sugar, and sugars have a lot of energy. The energy is going to be released during the cell respiration reactions. Okay, that energy will be used to build a molecule called ATP, which is the final goal product of cell respiration. We have not talked about ATP yet, but I will get to that soon in this presentation. Our cells require a very high energy molecule called ATP in order to continue to stay alive. ATP is going to be a very high energy molecule that is going to be produced due to the cell respiration reactions that occur. The energy that is released from the sugars will be used to make high energy ATP molecules. But since during the cell respiration molecules we broke down sugar instead of made sugar, it is going to be considered a catabolic type of reaction. Let's talk about reactions in terms of energy. So reactions that require energy reactions that require an input of energy are going to be called endergonic. They're also called endothermic. An example of a type of reaction that requires an input of energy would be photosynthesis. Photosynthesis requires a net input of light energy in order to begin the process of building sugars. Other examples of endergonic type of reactions are going to be any reactions that require energy. Usually these type of reactions are building larger, more complex molecules. For example, building proteins, building DNA. Let's go back to our example of photosynthesis to show what an endergonic reaction is. In order to show the example, we need to give the very basic equation of photosynthesis. During the process of photosynthesis, we start off with these two molecules. We studied these in class before. Hopefully you know what they are. The first molecule is called CO2. That's the molecular formula. That's going to be carbon dioxide with the addition of water plus a net input of energy. And that energy is going to be light energy since we're talking about photosynthesis. Once we have all those three factors, carbon dioxide, molecules, water, and a net input of energy, the plant can undergo the photosynthetic reactions and they end up producing these substances. Recall from the biochemistry lab that when the molecular formula is C6H12O6, that is a carbohydrate. In this case, we can call it a sugar. Our specific example that we're going to continue to use will be glucose. Please keep in mind that these are the reactants of our overall photosynthetic equation, and these are the products of photosynthesis. Various carbohydrates and the oxygen gas that we breathe. So let's not forget that glucose is a very high energy molecule and it ends up storing energy. Our cells love to absorb glucose because once glucose is inside of our cells, we can break glucose down to release the energy that was once stored in it. That energy is then going to be used as fuel for all the cellular activities that the cell needs to complete in order to stay alive. Energy that is stored, for example, the energy that is stored inside of the glucose molecule is going to be called potential energy. So the energy that's stored inside of glucose before that the cell breaks it down is considered potential energy. Anytime energy is stored, that's going to be called potential energy. Here are a few examples of potential energy. Glucose is going to be an example of potential energy. Any other sort of nutrients that you consume, like other types of carbohydrates besides glucose, or fats, or even the ATP molecule that we discussed earlier, are all examples of potential energy because energy is stored inside of these molecules until the cell goes to break them down to release the energy from them. So our first type of reaction was endergonic. Our second type of reaction in terms of energy flow is going to be called exergonic, which is also called exothermic. An example of an exergonic reaction is any time during a reaction where a very large burst of energy is released, that type of reaction would be considered an exergonic type of reaction. So an example of an exergonic reaction would be the breakdown of any type of nutrients. Our example is cell respiration is a process that breaks down sugar. Let's write an overall equation for cell respiration. Remember that the process of cell respiration is breaking down sugar. So we start off with our sugar. And in order to fully break down the sugar, oxygen will be useful for us. Oxygen is not going to be needed to break down sugar, but in this case, we will include oxygen because it is going to be the most efficient way to break down sugar. So we start off with sugar, which we know has a lot of energy. It is a very energy-rich molecule. And when we break down sugar, we are going to end up with carbon dioxide. We're going to end up with some water, but we will also end up with that big burst of energy that is released from sugar. That energy is going to be transferred to the energy that's going to be stored in our ATP molecules. So glucose and oxygen are going to be considered the reactants. They're going to be on the reactant side. And then over here are going to be the products. So one thing to note is that when energy, and that big burst of energy is on the product side, that is going to be considered an exergonic type of reaction. For our last example, that big burst of energy is on our reactant side. And anytime there is a big burst of energy, it's basically going to be a net input of energy. We're going to call that type of reaction endergonic. It turns out that all reactions require some initial energy. And we're going to call that type of energy activation energy. So let's go back to our molecule up here, which we're going to call sugar. Sugar is not broken down just instantaneously. Molecules need to be destabilized. So when we have a molecule like sugar, it's pretty stable. All the bonds are really stable within that molecule. So the bonds that are keeping the molecule together need to be destabilized just a little bit in order for the reaction to get going. The initial amount of energy that is needed for every single reaction is going to be called activation energy. And the type of activation energy that is usually used to begin all reactions or to start all reactions is heat energy. If you apply enough heat, that is going to end up destabilizing molecules enough to drive reactions. So heat energy is definitely one of the major forms of activation energy that is needed for all reactions to get started. However, living things are limited by the amount of heat energy that they can absorb. So living things basically have to have a workaround in order to get our reactions going. So living things, instead of absorbing enough heat in order to start some of the reactions that we need to happen in order to sustain our life, living things have to somehow decrease the amount of activation energy that would otherwise be needed to get a reaction started. Living things have something called catalysts, which basically reduce the amount of activation energy to get a reaction going. So as living things, our goal is to keep our reactions going at a rate which sustains life. However, we have a major problem in that all reactions require some sort of initial energy that we call activation energy in order to get the reactions going. And usually that energy is going to be heat. The problem is that living things can't just absorb heat endlessly. So living things have to have a workaround called a catalyst, which will reduce the activation energy that is needed and basically jumpstarts the reaction to go. We're going to talk more about the specific type of catalyst that living organisms need just pretty soon within the presentation. In living things, those catalysts are going to be called enzymes. Chapter 5 mentions the first law of thermodynamics, which we will talk about very briefly here. The first law of thermodynamics basically says energy can neither be created nor destroyed, only transformed during reactions. So energy is never created during a reaction. Energy is never destroyed during reaction. Energy is simply only transformed. We did give some examples about this earlier, but let's go ahead and redraw that sun. So here's the sun. Let's redraw our leaf right here. Right, here's my leaf. And remember that the sun emits something called light energy, as well as other types of energy. And the leaf is able to absorb various forms of light energy and transfer that energy to glucose molecules. Glucose represents chemical energy. Glucose is also an example of potential energy. Please do not forget that when light energy is transformed to chemical energy, there is always heat energy given off. Heat energy is given off during every single reaction. And the problem with heat energy is that living things cannot use it to do cellular work. So according to the first law of thermodynamics, energy isn't created or destroyed, it's only transformed. So our example basically is saying light energy is transformed into chemical energy during the photosynthetic reactions. So according to our first law of thermodynamics, we didn't create or destroy any energy. We only transform energy during reactions. So we start off with light energy, and that light energy is transformed to a different type of energy called chemical energy. Remember, there's always heat energy given off during every reaction. And this energy transformation happened due to the photosynthetic reactions. Cellular respiration is another example of the first law of thermodynamics. Make sure to read about cellular respiration in your online textbook. We have to mention our molecule called ATP. ATP is a very high energy molecule that cells use frequently. Cells have a very big demand for ATP, especially cells that are very energetic. So ATP stands for adenosine triphosphate. And it serves as immediate energy for cells. Let's first look at its structure, and then I will talk about the function a little bit more. I'm going to make a diagram of ATP. So this is the ATP molecule. The ATP molecule is going to be a modified nucleotide in structure. This is what ATP looks like. I know it looks a little complicated with this diagram, but ATP is actually a much simpler and smaller molecule compared to other molecules like proteins and DNA and lipids. And because the molecule is small, we can easily break it down to release the energy that's going to be available. In order to break ATP down, the energy is going to be released when the last bond between the last two phosphates are broken down. So remember that we have adenosine triphosphate, meaning there are 3 phosphate groups. So here's 1, 2, 3. And there is a lot of energy in this last bond between the last 2 phosphate groups. When the cell breaks that bond, this means the cell needs energy to perform work. When the cell breaks the bond, the molecule becomes ADP, adenosine diphosphate, because when we break that last phosphate group off, we're now only going to have 2 phosphate groups left. So we're left over with ADP plus an inorganic phosphate group roaming around in the cell. Okay, so ATP is the immediate source of energy for the cells. What about sugar? What about glucose? Glucose and sugars are wonderful sources of energy for the cells. But when we break down glucose or sugars during the cell respiration reactions, takes about 25 or 30 reactions to break down sugar. However, ATP can be broken down in one step. That means that ATP is going to be an immediate source of energy for the cells. It is a very quick burst of energy, and the amount of energy that is released is going to be usually more than enough to perform whatever specific types of cellular work that the cell demands at the time. Our cells are really good about recycling substances, so when our cells get some more sugar and break down sugar, the energy that's released from sugar is going to be used to put the ADP and the phosphate back together again to form the high energy ATP molecule. So when our cell gets food again, our cells receive sugars. Sugars are going to be broken down during a process called cell respiration, and the energy that is going to be released from the breakdown of sugar during cell respiration is going to be used to make high energy ATP molecules. So basically, these reactions continue to go backwards and forwards within our cell. Some examples of cellular work that requires energy from breaking down ATP molecules would be anytime our cells are building substances. For example, building proteins. Anytime our cells contract muscles. Anytime our cells are actively transporting substances. An example of active transport would be the sodium potassium pump discussed from chapter 5. So any type of cellular work in which we're building a larger molecule, or we're contracting a muscle fiber, okay, or we're actively transporting a substance will require the energy from ATP. In order to get the energy from ATP, the cell has to break ATP down, okay, and when ATP is broken down, the burst of energy is released to perform the work. Here are some examples of work. Okay, and then the cell basically takes the products and recycles it back in a continual cycle. All right, let's get to our term enzyme. Since our lab is about the pineapple enzymes, what the definition of an enzyme is that enzymes speed up reactions so that reactions occur at a rate which sustains life. Without enzymes, we would not be able to continue to live. So enzymes basically allow our cells to keep a pace of the reactions in order to keep our cells alive. Enzymes may catalyze, and that term catalyze means speed up. Enzymes may catalyze anabolic and catabolic type of reactions. Let's give an example of a catabolic type of reaction that an enzyme catalyzes. There is an enzyme called lactase which catalyzes or breaks down a substance which is called lactose. Lactose is often called milk sugar. It's going to be the sugars that are seen in milk or dairy products like ice cream. So this enzyme has a specific name. It's called lactase. And the enzyme catalyzes the hydrolysis, which, don't forget, that's a catabolic type of reaction, of a substance called lactose. The individuals that are lactose intolerant do not have the enzymes or the amount of enzymes that they would need in order to break down the lactose that they ingest. A supplement can be taken in order to ultimately supplement the enzymes so that the individual can enjoy lactose products. So notice the name of the enzyme is called lactase, and the substance that it breaks down is called lactose. So enzymes are often very specific about the types of substances that they catalyze. For example, lactase is an enzyme that will only catalyze lactose. Lactase will not be able to catalyze glucose. Lactase will not be able to catalyze sucrose. Lactase will not be able to catalyze any other substance besides lactose. So enzymes are very specific about the types of substances that they will catalyze. The next thing that we will do is talk about the steps of enzyme activity very briefly. I have a very basic diagram that I would like to explain. The first thing that we want to do is we want to point out which substance is the enzyme. So this big guy right here is going to be the enzyme. And before we write down some notes, let's just talk about what it does, and then I will add the notes. So the enzyme, which this is the enzyme, these are basically the steps involved in enzyme activity. The enzyme will ultimately intake the substances that it will catalyze. So these substances are going to be catalyzed. So they enter a very specific area on the enzyme called the active site. The active site is the region of an enzyme that will attract a specific substrate or substance into the area. These substances are going to be called substrate. In this case, the substrate is attractive to the active site of the enzyme, and it's usually due to basic chemistry. For example, positives and negatives, opposites attract. So the active site of the enzyme ultimately lures the substrate into the active site of the enzyme. And then what happens is the enzyme will twist the substrates just a little bit and destabilize the bonds within the substrates. This is going to jumpstart the reaction to occur, because please recall that molecules need to be a little destabilized in order to jumpstart any sort of reaction. Okay, so let's go ahead and list the steps of enzyme activity. The first step is substrate enter the active site of the enzyme. So here's step one. Substrate enters the active site of the enzyme. Step two is going to be induced fit. This is going to be when the enzyme kind of squeezes or destabilizes the substrate in order to jumpstart the reaction. And then it's pretty simple after that. What happens is the enzyme will release the products. That's step three. So basically, the enzymes release the products, and the products go out to do their job. So this guy right here, this label should be products. So just by looking at the type of reaction, you guys see that a substance enters, and then basically the substance is broken apart. So here's our substance, and then the substrate enters the active site of the enzyme. The enzyme twists the substrate just a little bit in order to destabilize the bonds within the substrate, and that's during the induced FET mechanism. And then what happens is the reaction occurs, which ultimately the reaction is going to occur to separate these two subunits from each other. So therefore, it looks to me like this substrate molecule was broken down. So therefore, in this example, the enzyme, catalyzed a catabolic reaction. And the reason I think that based on the diagram is because the products are no longer connected, they're separated from each other, meaning that this substance was broken down. So one thing that we want to make sure that we note is that the enzyme is unchanged. So the enzyme remains unchanged during this process, or I should say after the process has occurred. So once the enzyme releases the products, it is back to basically square one, where it's ready to accept more substrate molecules. So the enzyme can be reused over and over and over again as long as there is substrate available. Let's talk about factors that affect enzyme activity. A couple of factors can affect enzyme activity. For example, from our last picture or our last diagram, we saw that when substrate enters the active site of the enzyme then catalyzes the reaction. As long as there is substrate available, the enzyme will continue to catalyze reactions. So if there was no substrate, then the enzyme is not going to catalyze reactions. So the concentration or the amount of substrate could definitely affect enzyme activity. But some factors in the environment affect enzymes to the point where the actual structure of the enzyme itself is destroyed. So the term denatured means destroyed. Okay, and let's not forget that enzymes fall into the category of proteins for the most part. And proteins are really sensitive to their environment. So, for example, if you have a fever that is spiraling out of control, the first molecules that are going to be really affected by your body temperature going up is going to be proteins. They're very sensitive to various factors in the environment, including high temperatures. And the problem with a fever is that if it's spiraling out of control, the proteins are going to end up being destroyed, or what we call denatured, faster than the cells can replace them. So one factor that may affect enzyme activity will be temperature, especially high temperatures. So if you expose the enzyme to too high of a temperature, it could definitely destroy or denature that enzyme, therefore stopping all enzyme activity. The reason that the enzyme will stop functioning is because the high temperature will distort the overall shape of the enzyme, which is definitely going to affect the active site of the enzyme. So the enzyme will not be able to accept substrate. Another factor in the environment that could end up denaturing or affecting the enzyme activity will be the pH of the environment. So enzymes are very sensitive to the acid levels in their environment. Some enzymes function well in an acidic environment. Some enzymes like to function the best in a neutral type of environment. There are even certain enzymes that will function in a really basic type of environment. So it really depends on the type of enzyme that we're talking about. But let's go ahead and review some of the pH values. You will definitely need that for the Pineapple Enzyme Lab. The pH values for acidic substances are pH less than 7. Remember, anytime the pH value is less than 7, that's going to be considered acidic. Any pH values that are greater than 7 are going to be considered basic. Other factors in the environment that could end up denaturing is a really high salt environment. We do also have other factors in the environment which end up more or less regulating enzyme activity and not really destroying the enzyme. These substances are called inhibitors. And I want you to read about this section in your online textbook. The rest of these activities are on your own activities that are going to help you prepare for the pineapple enzyme. So you want to make sure to look in your lab manual to complete the rest of the questions, 12, 13, 14, and then on the next page you have number 15. Okay, in order to answer these questions, you need to read through that lab, which is called the Pineapple Enzyme Lab, in your lab manual.